everyone. In this section, we'll talk about the metabolic phase of pyruvate and how fructose, galactose, and mannose enter glycolytic pathway. The sequence of reactions from glucose to pyruvate is similar in the most organisms and most types of cells. In contrast, the fate of pyruvate is variable. Three reactions of pyruvate are of primary importance: conversion into ethanol, lactate, and carbon dioxide. The first two reactions are fermentations that take place in the absence of oxygen. Under aerobic conditions, the pyruvate is completely oxidized via the citric acid cycle to carbon dioxide and water. Firstly, let's see alcoholic fermentation. Ethanol is formed from pyruvate in yeast and several other microorganisms. The first step is the decarboxylation of pyruvate. This reaction is catalyzed by pyruvate decarboxylase. The second step is the reduction of acetaldehyde to ethanol by NADH. This reaction is catalyzed by alcohol dehydrogenase, and the NAD+ is regenerated. The conversion of glucose into ethanol is an example of alcoholic fermentation. This is the net result of this anaerobic process. Note that NAD+ and NADH do not appear in this equation. NADH generated by the oxidation of glyceraldehyde 3-phosphate is consumed in the reduction of acetaldehyde to ethanol. Thus, there is no net oxidation reduction in the conversion of glucose into ethanol. Secondly, lactic acid fermentation. Lactate is formed from pyruvate in a variety of microorganisms in a process called lactic acid fermentation. Certain types of skeletal muscles in most animals can also function anaerobically for short periods. The ATP needs rise faster than the ability of the body to provide oxygen to the muscle. The muscle functions anaerobically until fatigue sets in, which is caused in part by lactate buildup. This is the overall reaction in the conversion of glucose into lactate. As in alcoholic fermentation, there is no net oxidation reduction. The NADH formed in the oxidation of glyceraldehyde 3-phosphate is consumed in the reduction of pyruvate. Finally, let's see oxidative decarboxylation of pyruvate. Only a fraction of the energy of glucose is released in its anaerobic conversion into ethanol or lactate. Much more energy can be extracted aerobically. In the presence of oxygen, the most common situation in multicellular organisms and in many unicellular ones. Pyruvate is metabolized to carbon dioxide and water through the citric acid cycle and the electron transport chain, with oxygen serving as the final electron acceptor. The entry point to this oxidative pathway is acetyl coenzyme A, which is formed inside mitochondria by the oxidative decarboxylation of pyruvate. This reaction is catalyzed by the pyruvate dehydrogenase complex. Together with glucose, fructose, galactose, mannose are prominent metabolic fuels. After digestion, these monosaccharides enter the bloodstream, which carries them to various tissues. They are converted to glycolytic intermediates for further metabolism, and then metabolized by the glycolytic pathway. There are two pathways for the metabolism of fructose. One occurs in liver, and the other occurs in muscle and the adipose tissue. This dichotomy results from the different enzymes present in these tissues. Fructose metabolism in muscle differs little from that of glucose. Hexokinase phosphorylates fructose, yielding fructose 6-phosphate. The entry of fructose into glycolysis therefore involves only one reaction step. The main site of fructose metabolism is the liver, using the fructose 1-phosphate pathway. The first steps in the phosphorylation of the fructose to fructose 1-phosphate by fructokinase. Fructose 1-phosphate is then split into glyceraldehyde and dihydroxyacetyl pathway, an intermediate in glycolysis. This aldol cleavage is catalyzed by a specific fructose 1-phosphate aldolase. Glyceraldehyde is then phosphorylated to glyceraldehyde 3-phosphate, a glycolytic intermediate by Charles kinase. The entire pathway converting galactose to a glycolytic intermediate requires 4 reactions. Galactose is phosphorylated by ATP in a reaction catalyzed by galactokinase. Galactose 1-phosphate uridyl transferase transfers the uridyl group of UDP glucose to galactose 1-phosphate to yield glucose 1-phosphate and UDP galactose. Epimerase converts UDP galactose back to UDP glucose. Glucose 1-phosphate is converted to the glycolytic intermediate, glucose 6-phosphate, by the action of phosphoglucomutase. Mannose enters the glycolytic pathway after its conversion to fructose 6-phosphate via a 2-reaction pathway. Hexokinase recognizes mannose and converts it to mannose 6-phosphate. Phosphomannose isomerase then converts these aldols to the glycolytic intermediate fructose 6-phosphate. Okay, that's all. Thank you.